Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
In the grand intellectual theatre of the late 20th century, few publications signalled a more profound or unexpected paradigmatic shift than Leonard Adleman's 1995 paper in the journal Science, titled Molecular Computation of Solutions to Combinatorial Problems. This work represented an epistemological rupture, a moment where the abstract, logical universe of computer science collided with the messy, tangible reality of molecular biology. Adleman, a distinguished theoretical computer scientist already known for his foundational contribution to RSA cryptography, ventured into the wet laboratory and emerged with something extraordinary, a demonstration that the very molecules of life, DNA, could be harnessed to solve a complex computational problem. His experiment was far more than a mere novelty. It was the progenitor of an entire field, DNA computing, and a powerful physical argument that the substrate of computation is a universal principle, unbound by the silicon hegemony that had defined the digital age. This essay will explore the intellectual genesis of Adleman's work, dissect its brilliantly conceived methodology, trace its immediate and long-term legacy, and evaluate the critical conversations it initiated, revealing it as a landmark achievement that fundamentally reimagined the boundaries of information pro. The conceptual groundwork for Adleman's experiment was laid decades prior, within two seemingly disparate intellectual traditions. In computer science, the theory of computational complexity had elegantly classified problems into hierarchies of difficulty. Among the most notorious of these were the problems in the class known as non-deterministic polynomial time, or NP. A particular challenge within this class, the Hamiltonian Path Problem, served as a benchmark for computational intractability. The problem asks for a path through a network of cities that visits each city exactly once. While verifying a proposed solution is simple, finding one for a large network requires a search of possibilities that grows exponentially, quickly overwhelming even the most powerful electronic computers. This limitation of conventional, sequential processing defined a clear frontier. Concurrently, the world of physics and biology was undergoing its own revolutions. Richard Feynman's 1959 lecture, "There's Plenty of Room at the Bottom," had planted the seed of computation at the molecular scale, a prophetic vision of manipulating individual atoms as bits. By the early 1990s, molecular biology had developed a sophisticated toolkit capable of realizing such manipulation. Techniques like the polymerase chain reaction, or PCR, for amplifying specific DNA sequences, DNA ligase for stitching strands together, and gel electrophoresis for sorting molecules by size, were standard laboratory practice. Adleman's genius lay in his conceptual synthesis. He perceived these biological tools as a set of computational primitives, a means to execute an algorithm whose power derived from a fundamentally different. The specific problem Adleman chose was a small instance of the directed Hamiltonian Path Problem, defined on a graph of 7 nodes, representing cities, connected by a series of one-way edges, representing flights. His first and most critical innovation was the encoding scheme, a translation from the abstract language of graph theory into the chemical language of genetics. Each of the 7 cities was assigned a unique random sequence of 20 DNA bases. These single-stranded oligonucleotides functioned as the nodes. Each directed edge, for example a flight from city 2 to city 3, was then represented by a complementary DNA strand. This flight strand was designed to bind to the second half of the city 2 sequence and the first half of the city 3 sequence, effectively acting as a molecular bridge. The Watson-Crick base pairing rules, where adenine pairs with thymine and guanine with cytosine, provided the specific, predictable binding mechanism. By creating a literal test tube containing trillions of copies of all the city and flight molecules, Adleman established the initial conditions for his computation. This soup of DNA was the physical embodiment of the problem space, a liquid medium containing all the necessary components for constructing potential solutions. The computational process itself was a sequence of meticulously orchestrated biochemical reactions, each step performing a logical operation. The first step was one of vast, anarchic creation. By adding DNA ligase to the mixture, Adleman catalyzed the formation of long DNA strands as the flight connectors randomly annealed to and joined the city sequences. This single chemical reaction generated a massive library of molecules representing all possible paths through the graph, a display of parallelism on a scale unattainable by any electronic device. The test tube at this stage contained a chaotic jumble of valid and invalid paths, long and short paths, and circular routes. The subsequent steps of the algorithm were a process of systematic, logical filtration. To isolate paths that began at the designated start city and finished at the end city, Adleman employed PCR, using primers corresponding to the known sequences of the start and end cities. he selectively amplified only those molecules that had the correct termini. This action was equivalent to a massive parallel search for all paths with the correct beginning and end. Following this initial selection, the next challenge was to isolate paths of the correct length. A valid Hamiltonian path in a 7-node graph must contain exactly 7 city sequences, corresponding to a DNA molecule of 140 base pairs. Adleman used gel electrophoresis, a standard technique that separates molecules by size as they migrate through a gel under an electric field. This step physically filtered the solution, allowing him to extract only those molecules of the target length, effectively discarding all paths that visited too few or too many cities. The final and most subtle step was to ensure that the remaining candidate paths visited every single one of the 7 cities. For this, Adleman devised an ingenious iterative process of affinity purification. He would take the solution of candidate paths and expose it to magnetic beads coated with DNA complementary to the sequence of City 2. Only those molecules containing the sequence for City 2 would bind to the beads. He could then magnetically separate these molecules, wash away the rest, and release the captured strands. He repeated this exact process sequentially for City 3, City 4, City 5, and City 6. Any molecule that survived this entire gauntlet of purification steps necessarily contained every city in the correct path. As a final act of readout, he sequenced the remaining DNA, and the order of the bases revealed the correct path, the solution to the problem. He had, with a series of chemical manipulations, computed the publication of this result in November 1994 sent shockwaves through the scientific community. It was a stunning proof of concept, demonstrating that computation was a physical process whose principles could be realized in biological substrates. The immediate impact was the birth of a new interdisciplinary field, molecular computing. The paper's core philosophical contribution was its vivid illustration of the power of massive parallelism. In that single test tube, roughly 10 to the power of 14 molecules were operating in parallel, a computational throughput that dwarfed the capabilities of the world's supercomputers. While each individual biochemical operation was slow, the sheer number of simultaneous calculations enabled the rapid exploration of a complex combinatorial search space. This offered a compelling alternative for a specific class of problems where the ability to check many possibilities at once is paramount. The work inspired a generation of scientists at the intersection of computer science, chemistry, nanotechnology, and biology to think about information in a new light. In the years following Adleman's experiment, the field he founded evolved and matured. Researchers recognized that solving single instances of the Hamiltonian Path Problem was perhaps a practical dead end. Instead, they began to generalize the principles of molecular programming. At the California Institute of Technology, Eric Winfrey, directly inspired by Adleman, developed the concept of algorithmic self-assembly using DNA tiles. These were specifically designed DNA nanostructures that could be programmed to assemble into larger, more complex patterns, like a Sierpinski triangle, effectively performing a computation as they grew. This shifted the focus from one-off calculations to creating programmable matter. Paul Rothermund, also at Caltech, extended this idea with his invention of DNA origami, a method for folding a long single strand of DNA into an arbitrary two-dimensional shape using shorter staple strands. These advances transformed DNA from a simple computational medium into a sophisticated nanoscale construction material. Elsewhere, research groups like that of Ehud Shapiro at the Weizmann Institute of Science developed autonomous molecular automata, tiny DNA-based machines that could operate inside a test tube, and even within living cells, to sense for disease markers and trigger a therapeutic response. This represented a move from in vitro computation to creating programmable biological agents, a direct intellectual lineage from Adleman's initial demonstration. Despite the initial excitement and subsequent innovations, the field of molecular computing also faced sober-minded critique and significant practical hurdles. The primary limitation, acknowledged by Adleman himself, is one of scalability. While his 7-city problem was manageable, the amount of DNA required to represent the problem scales exponentially with the number of nodes. Solving the Hamiltonian Path Problem for a mere 200 cities would, by some estimates, require a mass of DNA greater than the weight of the Earth. This makes the approach impractical for tackling the large-scale problems where it would be most useful. Furthermore, the biochemical operations are slow, prone to errors, and require significant manual intervention, a stark contrast to the speed, reliability, and automation of silicon-based processors. This reality has tempered the original vision of DNA computers replacing conventional ones. A reception thus evolved from unbridled optimism to a more nuanced appreciation of molecular computing's strengths. The field has found its niche where its unique advantages align with the problem domain in massively parallel search for specialized problems. in the construction of nanoscale objects, early interface with biological systems for applications like biosensing and smart therapeutics, where a biocompatible, programmable molecule is the ideal. In conclusion, Leonard Adleman's 1995 paper stands as a seminal work of modern science, a testament to the power of interdisciplinary insight. It was an elegant and audacious piece of research that used the fundamental components of life to solve a problem born from the abstract logic of mathematics, while the paradigm he introduced has evolved away from the goal of building a general-purpose bimolecular computer. Its intellectual legacy is undeniable and profound. The paper demonstrated that computation is a physical process, its principles expressible in chemistry as well as in electronics. It launched the field of molecular programming and laid the conceptual foundation for nanotechnological triumphs like DNA, origami, and the development of molecular robots. Adleman took the abstract idea of a Turing machine and gave it a startlingly new, tangible form in a drop of water. He revealed that the same double helix that encodes the instructions for life could also be programmed to reason, calculate, and solve. In doing so, he forever expanded our definition of what a computer can be, opening a rich and ongoing dialogue between the science of information and the science of life itself.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.